Thank you for the invitation to talk at this GenTAC Aortic Summit. I'll be updating you on the genes for heritable thoracic aortic disease and giving you an update on the data from the Montalcino Aortic Consortium. Here's the list of current heritable thoracic aortic disease genes. These are genes that when they are altered or mutated confer a very high risk for dissections. The ones bolded in black are those that are confirmed to cause disease and can be used clinically. The rest of them are awaiting additional data. Despite having this long list of genes in our cohort, we still can only explain the disease in about 35 to 40% of the cohort. So we still have more genes to be identified. So what are these genes telling us about signaling? Well, we have genes that disrupt the extracellular matrix, the TGF-beta signaling, the ability of the smooth muscle cell to contract. And then down here, we have some new genes that are disrupting potassium channels in smooth muscle cells. And I'll talk more about those in a minute. So let me just focus first on what these genes in the extracellular matrix and those involved in smooth muscle contraction may be telling us. So these genes encode proteins involved in the structure in the aortic wall called the elastin contractile unit. And this is where extensions from elastin, shown here in black, reach down in an oblique manner and attach up to contractile units in the smooth muscle cells. And those genes in the extracellular matrix and contractile units disrupt components of this structure, leading us and others to suggest that these genes are disrupting the ability of the smooth muscle cells to mechanosense. And that with that disruption, the pathways that lead to thoracic aortic disease are activated. And so many studies are coming out that address the role of mechanosensing in the aorta. I just also want to mention that those genes involved in TGF-beta signaling are affecting genes along the canonical TGF-beta pathway, and uniformly they are causing loss of function. And so we know from these data that loss of TGF-beta signaling is a primary driver of heritable thoracic aortic disease. Now I want to mention these two channel genes. HCN4 is a known gene for bradycardia and some cardiac abnormalities. And there are a few variants that my lab and other labs have validated to cause thoracic aortic disease. And that data is always published. We have new data on the potassium channel, KCN5A, and I referred you to Dr. Glow's abstract for further for additional information. I'm just going to briefly review the data here. We pulled this gene out of from the exome data because we found it in two unrelated in the same variant here, this arginine 234 to serine, in two unaffected patients with thoracic aortic disease, one from our heritable thoracic aortic disease cohort and one from our early onset aortic dissection cohort. And this variant was not in any of the genome data sequencing databases. We went on to pull out other variants. KCN5A is a gene already established to cause atrial fibrillation, and we hit some of our patients with thoracic aortic disease had variants that had already been identified in patients with AFib, shown here in blue. We went on to show that some of that in two small families, these variants did segregate with disease. In our cohort of individuals with KCN5 rare variants, all of them had thoracic aortic disease, and 1/3 of them also had AFib. And here's where those variants are located. The blue indicates the variants already identified for AFib. Our variants in thoracic aortic disease patients are shown in red. They found in the intracellular cytoplasmic domain or in the transmembrane domains for this gated channel. And we went on to show with collaborators that these variants do disrupt the function of this channel. So this provides limited to moderate data that this is another HTAD gene, but it will need further verification before we can take it into to use for clinical trials. I also want to mention that we have accumulated data on a gene called PLEKHOZ over the last few years, and our data now indicated that this is the first gene that disrupts non-canonical TGF-beta signaling, this pathway over here. And so this is a very complicated story, so look for that data, which will hopefully be submitted and published soon. I also want to mention that there's 2 papers in bioRxiv that use the UK Biobank to look for genetic variants associated with aortic size. These were basically very large GWAS studies that pulled out hundreds of loci with genetic variants that predisposed, that were associated with aortic size in the general population. They did hit 2 of our previous GWAS hits in EPN1 and ULK4. And they also pulled out 2 known HTAD genes, EPN1, and a new gene, THSD4. We have gone on to look at the over 100 genes that they pulled out for aortic size, and we only have limited data that about 4 of those genes may confer a risk for heritable thoracic aortic disease. And then finally, I want to update you on where we are with the Montalcino Aortic Consortium. We have focused in looking at the clinical. The Montalcino Aortic Consortium has been used to collect clinical data on all patients that have variants in HTAD genes, with the exception of EPN1. We're only collecting limited data on patients with EPN1 mutations. I'll be talking about our data associated with the genes in red, but we're collecting data for all of these genes. Here is the Aortic Consortium. This involves aortic centers from around the world, and we're happy to have funding from a number of agencies. And please be we're happy if you refer our patients for the MAC Consortium, or you're welcome as aortic investigators to join MAC if you see these patients. Just let us know. And I want to present data on those genes, those 7 genes that I mentioned. Look for a publication soon on these genes. Here's the just a really quick snapshot of the paper that is comparing all these 7 genes side by side. And once again, this is just one piece of data from this paper. This is the age of an aortic event, defined as surgical repair of an aneurysm or a type A or type B dissection, versus the age, the number of people that have had an aortic event. So this is for those Loeys-Dietz syndrome genes, the TGF-beta genes listed here. And these are for the genes that are involved in smooth muscle contraction. So if we look at the age at aortic event, you can see that looking across the graphs for the SMAD3, TGFB2, TGFBR1 and 2, that there's significant difference in the age of onset of aortic events, with SMAD3 being significantly later than TGFBR2 and TGFB are 1. And then this is the dashed line is TGFB2. Once again, there's also a significant difference in the smooth muscle contractile genes, with PRKGB1 being the earliest onset and actually slightly earlier onset in terms of dissection than even TGFB2, and myosin light chain kinase being the latest onset. And then if we look at the age of onset, you can see that TGFB1 and 2 are associated with childhood onset, and whereas TGFB2 and SMAD3 are not, and that ACTA2 is also associated with child onset disease. So these data have really significant implications for the way we manage and screen these patients. So be looking for this paper soon also. So I want to thank all my collaborators and sources of funding, and in particular, all the people in the lab. So thank you once again.